So I have dual mode in this next session. I'm going to introduce the first of a pair of speakers who have been working together for the last 15 years or so, and that's Jeff Volek and myself. And just to give you a 30-second sense of how we met, our first meeting occurred when Jeff crashed a CME course in San Diego. He managed to sneak in and sat down next to a guy in the back row and said, do you know who Steve Phinney is? Is he supposed to speak soon? And I said, yeah, it's me. I don't remember that was Jeff. I just remember the incident because after I gave my talk, he and another guy, who both of them at that point were competitive power lifters, got me in a corner and asked me a bunch of questions. And that was 1996, I think. But in 2003, Jeff and I reconnected at a nutrition conference in San Francisco. I walked by a poster that had work on a ketogenic diet and body composition in males with metabolic syndrome. And I looked at that and said, wow, that's amazing. And then I noticed that it referenced a couple of my papers from back when the dinosaurs walked the earth. And I met the co-author, and the relationship for the last 15 years has been, for me, kind of a career resuscitation and just a true gift. So what I want to do is introduce Jeff at this point. As you know, he's a professor here at Ohio State University and an international expert in the topic. And we will be talking about the emerging science of nutritional ketosis.
Okay, well, that was just really an excellent talk, John, and it kind of struck me that this tool of a ketogenic diet, from a therapeutic perspective, it's really the opposite of how you think of drugs being developed, where you start with the chemistry, you know what the formula is, and you know what the target is, and you do animal studies, and then you take it into various phases of human trials, and we're kind of doing the opposite here with ketogenic diets. I mean, we know they work in humans. We've known for a long time they work to treat seizures and even diabetes, and now we're backfilling that with some of the molecular mechanisms. So, I mean, it's just fantastic work you're doing, and it's a lot of still important questions to address related to how we wield this tool and how it works, but we know it works in many ways. So I put this historic slide together just to emphasize the point that in many ways, from my perspective anyway, we've been living in the dark ages when it comes to our diet policy and nutrition. We're ignoring a lot of relevant science. But in particular, if we look at the metabolite, ketones as a metabolite, they've been maligned more than any other metabolite that I can think of. And this is really unfortunate. If you go back, and on the top here, I've kind of got a lot of the negative factors that have contributed to this connotation of ketones being negative. It really started with their discovery. They were discovered back in the middle of the 19th century in the urine of uncontrolled diabetics. And that negative connotation has really stuck, as labeling these as a harbinger for metabolic problems. And then, you kind of fast forward, insulin was discovered in 1920s by Frederick Banting. And, and that prior to that time, ketogenic diets were actually used to treat diabetics. But with the discovery of insulin, and this was a life-saving discovery, super important, but it also allowed us to continue what at the time was we started to embrace agricultural carbohydrates. And then, of course, you have Ansel Keys coming on the scene with the diet heart hypothesis, further promoting low-fat, high-carbohydrate diets, which led to the first U.S. dietary guidelines in 1980, followed by the surge in obesity and diabetes. And we're fighting this negative perception around ketones which are fat-derived metabolites. But what's going on in the bottom of this slide are a lot of positive things. You can go back pretty far in history and read about the use of ketogenic diets. You have William Banting, different Banting, writing the Letter of Corpulence back around the same time the ketones were discovered. And you do have ketogenic diets being successfully used by a lot of physicians to manage both diabetes and epilepsy back in the early part of the 20th century. You have Stefansson's work, which I think Steve's going to talk a little bit more about. He literally locked himself up and consumed a ketogenic diet to prove that it wasn't harmful. But notably, you have George Cahill's pioneering studies at Harvard throughout the 60s and 70s, really documenting the underlying metabolism and physiology of ketones during starvation. And, I still go back and read these papers in the 60s. They were just elegant experiments documenting how starvation ketosis plays out physiologically. And you've got Dr. Atkins coming on the scene in the 70s, who popularized the Atkins Diet, which is still very prominent today. And we're fortunate to have Atkins as a sponsor here. And so if you look throughout most of the 70s, 80s, and 90s, there's very little research done on ketogenic diets. It was a toxic topic to be involved with one notable exception, and that's Steve Phinney's work. And so he published some really seminal work on keto adaptation and ketogenic diets in the 80s. And I think, these for me, these were just like reading word out of the gospel. I mean, these were studies that existed when no other people were doing studies. And so that really inspired me to, think that I was thinking along the right direction. And I started to do work in the late 90s, and I think several other people started to do work as well, including Eric Westman, and it's just grown from that time to now. Over the last 15 years, I would say we're entering the golden age of ketones, where we have all these new discoveries, which we'll hear about today, on mechanisms of ketones, on applications of ketogenic diets. So welcome to the golden age of ketones. I just want to mention briefly what my team does here at OSU. I'm really fortunate to have an incredible group of doctoral students, master's students, undergraduates, and other team players that contribute to a really exciting environment where we're trying to advance the knowledge and application of ketogenic diets. Fundamental to what we do is feeding studies. So we've established infrastructure for doing controlled feeding studies, ketogenic studies, and trying to apply that in as many different populations, ranging from clinical work, a big focus on cancer. You'll hear Parker Hyde talk Friday morning, but we're also studying at the other end of the spectrum, military performance and how we can enhance the soldiers' health and readiness and resilience and performance, and a lot of underlying physiology, including muscle adaptations, cardiac adaptations, etc. So we have, I think, about a half a dozen posters on display that my students will be standing next to this afternoon or this evening at our poster session. That'll give you a little snapshot of some of the work that we're currently involved with. So wanted to just introduce a couple terms, and you've heard these before, but I think it's worth emphasizing. When we talk about ketosis, understanding concentration is really fundamental. If you're eating a normal mixed diet, your ketones are suppressed. So your concentration of ketones in your circulating blood is, for most people, under 0.2 millimolar. When you restrict carbohydrates, for most people under 50 grams a day, you normalize ketones into the range of nutritional ketosis, which we've sort of arbitrarily defined as between 0.5 and up to 3, 4. Occasionally, you can transiently get a little higher than that. So that's an entire order of magnitude higher than you would have in the carb-fed state, but an entire order of magnitude lower than what you see in ketoacidosis. So you've got really, you can suppress ketones down to 0.01 millimolar, all the way up to 10 millimolar in ketoacidosis. That's 3 orders of magnitude different in this particular metabolite. And when we're eating a ketogenic diet that's well formulated, you're simply normalizing ketones into this range of nutritional ketosis where we're increasingly learning we have broad spectrum health benefits. And when you do that over a sustained period of time, that really encompasses what the term keto adaptation means. And we're just scratching the surface on understanding the various ways in which tissues and organs adapt to being in sustained ketosis. But fundamentally, you're enhancing reliance on fatty acid oxidation and ketosis. And we've documented this in different populations. You basically double your rate of fat oxidation when you're in ketosis. It's true if you're a type 2 diabetic with insulin resistance or if you're an elite ultra-endurance athlete. The absolute numbers are different, but relatively speaking, you double your rate of fat burning. And that, I think, is important for a variety of reasons. So keto adaptation is really a perfectly natural process that, as humans, we have evolved to have this very elegant, sophisticated system of being able to not just survive, but thrive in the context of very little carbohydrate. And you could argue from an evolutionary perspective, at least intermittent ketosis was the normal state before the agricultural revolution, which started maybe 10,000 years ago. And that's less than 2% of human history. So I just very briefly wanted to show you a little bit of data from experiments we've done over the years, and we've done a lot of prospective studies. This is just one in particular in people with metabolic syndrome or prediabetes. So these were individuals randomized to either a low carbohydrate or a ketogenic diet, and right off the bat we see people randomized to a ketogenic diet lose more weight without specifically instructing them to restrict calories. So these are ad libitum ketogenic diets, and we see this time and time again at obese people that follow a well-formulated ketogenic diet don't need to count calories, that they naturally restrict energy. And you can see the individual responses on the right where the average weight loss in the ketogenic group is higher than any individual person on the low-fat diet. And we've done a lot of work characterizing the insulin-resistant phenotype, if you will, around responses to ketogenic diets, especially around dyslipidemia. And we see that cholesterol profiles improve. There's nothing more potent than a ketogenic diet to lower triglycerides. And it also consistently raises HDL cholesterol and improves the particle distribution. And you'll hear from Ron Krause later today. But it does have a variable response on the LDL cholesterol concentration. But independent of that, you consistently see decreases in the small LDL particles. And there's increasing evidence that they are the most atherogenic particles circulating. We see improvements in glucose and insulin and measures of insulin sensitivity, as well as improvements in hormone sensitivity. So we see improvements in or greater decreases in leptin. We've also done similar work with thyroid hormones, yet we see no change in the function of these hormones, so no signs or symptoms of hypothyroidism, for example, even though T3 is dropping. So that leads us to believe that we're enhancing sensitivity to these hormones, perhaps at the receptor level and signaling level. We've also measured saturated fat levels in many studies now, not just this study, And a consistent finding is that despite consuming 2 to 3, sometimes even 4 times as much saturated fat on a ketogenic diet, that levels in the blood actually go down or stay the same. And this has led us to the concept that you are not what you eat, you are what you save from what you eat. And this kind of piggybacks off on the enhanced fat oxidation. When you're eating saturated fat on a ketogenic diet, you're promptly oxidizing that incoming saturated fat and converting it to CO2 and water. Saturated fat is, in fact, a preferred fuel when you're in ketosis. And this is very important because elevated levels of saturated fat in the circulation and in membranes is consistently associated with higher risk for heart disease, diabetes, even some types of cancer. And you heard from John that there were some anti-inflammatory effects in the animals fed ketogenic diets. We see the same consistent effect in human trials. So half the inflammatory markers we measured in this experiment were significantly decreased in the group fed a ketogenic diet compared to a low-fat diet. And we're seeing this in some of our ongoing trials, including patients with type 2 diabetes, that there's a potent inflammatory mechanism. And you'll hear from Dom D'Agostino after this talk about some of the mechanisms that may be at play here. So what we've learned over many studies now, over many years, is that a ketogenic diet essentially reverses all the signs and symptoms of metabolic syndrome and this insulin-resistant phenotype. And it's led me to kind of view this on a continuum where the more carbs you eat, the more you have a higher likelihood of developing the insulin-resistant phenotype, and if that progresses, type 2 diabetes. The more you restrict carbohydrates, especially down to levels that induce ketosis, the more you express this keto-adapted phenotype, which is a non-diabetic phenotype. And it's driven by carbs in the diet. Everybody has a slightly different carb tolerance, and it changes over the lifespan and perhaps other environmental factors. But this relationship between carbs and manifestation of insulin-resistant phenotype versus keto-adapted phenotype holds true. So I thought it would be important, because I'm not sure it will be addressed by other speakers, to talk about the idea of a well-formulated ketogenic diet. I really appreciate John mentioning some of the nuances around, optimizing the diet for the animals, and it's certainly very relevant for the human as well. And we have some understanding of how to best formulate and implement the diet to maximize safety, effectiveness, pleasure and satisfaction with the diet, and most importantly, sustainability. And so on that note, I will just say the diet's anything from a burden or sacrifice from my perspective, and I think many of you in the room would concur that it's in fact a very pleasurable way to eat and enjoy food. But I'm going to invite Steve to the stage to expand on that a bit and talk to you about some of the characteristics of a well-formulated ketogenic diet.
So one of the concepts that Jeff and I wanted to do was mix in not just the elegant basic science and mechanisms here and pathways, but also to provide some guidance into how this translates into something useful in the real world. And you'll hear a number of presentations on different disease states and such. But what I wanted to deal with is something I've struggled with for a number of decades, and that is, okay, we can do this in the lab, we can do it in the metabolic ward. What happens when we take this out in the real world? And one of the first places is to define, we use this term well-formulated ketogenic diet, but what is that? What are the confines of that? And probably the best visual representation we've come up with is to differentiate this from the standard American diet, what the average American and most people in the developed world eat. A better rendition of that health-wise is moving to a Mediterranean diet with less carbs, a different mix of fats. And then there is the Paleo Diet based on our understanding of what our distant ancestors ate, and so that takes the evolutionary perspective of what were our genes over the last 2 million years exposed to, and that's thought to be higher in protein, lower in carbohydrate, and it's typically called low carb, but it's still in the 20 to 30% range. And so I really thank Professor Ramsey to put some of his mice onto a mouse paleo diet and look at the effects on that. And there are some benefits, but there, at least in the mouse model, there is a very significant distinction between a higher protein and significant carb level, which will keep ketones under 0.3, fasted ketones under 0.3 millimolar in most humans unless they're doing high intensity, high volume exercise. So to achieve nutritional ketosis really provides this narrow island of, we think of it as an island of safety. Typically, it's under 10% of daily energy intake relative to one's daily expenditure. So here we're not talking about macros in terms of what's on your plate, but if you're burning 2,000 calories a day, you have to get typically under 20 or 10% of that, which would be about 50 grams of carbs to be on this island of safety. And again, if you take your protein intake too high, that protein intake begins to suppress ketogenesis. So it has to be a moderate protein, very low but not zero carbohydrate intake. And then if you do the math on that, if it's 10% or less carbs and it's 20% or less protein, it's, oh my gosh, what your body has to burn each day is more than 80% of its energy is fat. And that's really daunting. How do we do that? And Jeff showed you some pictures, and that's compatible with a well-formulated ketogenic diet. So how does one do that? And thinking, again, in terms of about a 2,000 calorie per day intake. And again, we don't specify calories. As Jeff said, the fat is eaten to satiety. We let the natural, highly evolved human instincts is when you take the carbs down, get that insulin signaling down, let natural instincts drive fat intake. But if we restrict carbs to anywhere from the 25 or 30 grams per day total intake, up to 50 or so, what can you have? And you can see that recognize that there is some carbohydrate in protein foods. When an animal is slaughtered, there's glycogen in the muscle, and that's bioavailable carbohydrate, but it's a small amount. So eating a moderate protein intake, that protein-based carbs might be 5 to 10 grams per day. If you eat 3 to 5 servings of non-starchy vegetables per day, depending on your choices, that puts you in a 10 to 15 gram range. If you give people 1 to 2 ounces of nuts and seeds per day, that's again 5 to 10 grams. At the higher end from 20 up to 50 grams per day, if you give people 4 ounces of berry fruit per day, that's again a 5 to 10 gram of carbohydrate intake. And then there's things in sauces and such as that. So you can see that one can stay in the 30 to 50 gram range and have a range of choices. And again, from the pictures here, realize that there are quite a bit of protein-containing foods here and there and cheeses, but the vast majority of the calories here are coming from fat. And it doesn't mean that the person has to eat all their daily energy need as fat, because we let them let their instincts dictate what their intake is, and they can choose from a wide range of luscious foods and condiments. And so, when we say, well, we take away most of your starches and we take away much of the sweets that we typically eat, but we give you savory and we give you unctuous, it's a trade-off. We also give people who have severe metabolic impairment, such as type 2 diabetes, metabolic syndrome, and other inflammatory-based diseases, we also give them not just a technical improvement in metabolic health, but we give them a subjective response. Again, when somebody with type 2 diabetes who's taking 100 units of insulin a day gets off 100 units of insulin a day, that's a very tangible perceived benefit. And we can do that when we get the composition right and we individualize it to the metabolic needs of each person rather than having a single formula cookie cutter approach. So this is variable, individualized, but again, the key here is keeping protein moderate and carbohydrate restricted. So one of the questions that I'm always frequently asked is, so what macro should I be eating? And I always push back and say, well, we don't do macros. We define carbohydrate on an individual tolerance basis, and the best way to determine your carbohydrate tolerance, if you want to be in nutritional ketosis, is you get a fingerstick glucometer with a ketone testing and measure your ketones. And that is now a very robust and accurate technology, and now we have multiple vendors who are coming out with provenly accurate devices. So as Jeff showed you when he did the study with Cassandra Forsyth and took people with metabolic syndrome and put them on a well-formulated ketogenic diet, and they ate to satiety, initially they underate calories. So in this, what we call induction phase, if they're eating 90 grams of protein and 30 grams of carbohydrate, that'd be 120 calories of carb and 360 calories of protein. And then when they ate fat to satiety, they were under-eating total calories. And so the difference between their expenditure and their intake was basically unseen, quote, dietary fat, but the dietary fat was coming from internal reserves. And as people typically follow this over many months, the weight loss continues, but it tapers, and eventually people will naturally come to a new steady state. And that is a combination of increasing dietary fat, so their need to meet satiety goes up. And also, if somebody loses 40 pounds, their resting metabolism is going to come down somewhat. And that's a reality. And that's a normal physiological response. That's not a toxic effect of the diet. It's just coming from carrying an extra burden of fat, which has metabolic requirements both at rest and during exercise. And people reach a new steady state. And so the key here, again, is we don't dictate calories and we don't count calories. We basically rely, and this is not in everyone, but in most people, their natural instincts are to underconsume through a period of time to weight loss, and then they reach what is a new steady state. And the important point here is that across these phases, our dietary instruction doesn't change from day one till the end. They may increase their carbohydrates slightly if they can maintain ketosis with a little bit greater intake of carbohydrate, which gives them a broader range of food choices. Which means they can have a caprese salad with 4 ounces of tomato, and maybe some berries at breakfast in the same day and maintain that metabolic beneficial state. So I want to begin to wrap up with what Jeff and I have, working with Dr. Brooke Bailey, who's in the audience, have come up with how do we define a well-formulated ketogenic diet. And we came up with basically 10 principles, and these are posted on a blog post that we put up recently at our blog at Verta Health. And the most obvious one is that until proven otherwise, sustained nutritional ketosis in humans at least appears to have better clinical response than intermittent ketosis in terms of degree of weight loss and improvement in manifestations of insulin resistance. So, it seems like a no-brainer, but it's still a point of contention, and there's still a lot of work to be done, whether intermittent ketosis or continuous ketosis is the most beneficial. But the greater the degree of insulin resistance our experience is, the better the response will be if somebody sustains nutritional ketosis. The next is that it needs to maintain lean body mass and function. And again, you've seen that elegantly in the mouse model that Professor Ramsey presented to you. And Jeff has shown that in his research, that we can take people on a well-formulated ketogenic diet, eating to satiety, not overeating protein, and yet they either maintain or increase lean body mass. So again, this idea that it's a muscle waste, it has to be a very high protein diet or waste muscle is inaccurate. Probably the most difficult one to convince people that it is important is electrolyte and mineral management. We assume that people will just naturally eat enough minerals and electrolytes to maintain their needs, but when someone enters a state of nutritional ketosis, It changes the kidney's functioning of sodium. And that's been known for, since the 1950s. It's called the natriuresis of fasting, natrium being the Latin term for sodium, that renal sodium excretion increases when one's in nutritional ketosis. And to put it as simply as I can, if you combine a ketogenic diet with a sodium-restricted diet in somebody who doesn't have obvious need for sodium restriction, that is hypertension or congestive heart failure, you'll cause a decreased circulating volume, and that leads to a whole panoply of symptoms and side effects that are unnecessary, and those are oftentimes called the keto flu. And that's not keto flu. That's the inadequacy of an essential nutrient called sodium to meet physiological needs. Again, as I've emphasized in the previous slide, fat needs to provide the majority of dietary calories in all phases. And again, we can discuss, well, what about in the early phase? Can we cut fat way back and have people lose more weight? And I'm going to address that in the next slide. A lot of people like to count calories, and if they want to, they can, but we don't prescribe calories, and we don't tell people to purposely restrict in most circumstances, because that's not something that humans are designed to do for years and decades. So most calorie-restricted diets fail within the first 6 months of the first year. And again, our predilection is this should be mostly or wholly composed of whole foods or real foods. That manufactured foods may have some roles in some circumstances, but what people are going to eat for the duration of their lifetime is going to be predominantly real foods that are purchased in normal pathways of grocery stores, or if you get a box delivered to your door or whatever, it should be composed of real food. Now, here I want to be a little bit provocative, but also science-based. And I will say that in my experience, having done short-term, very low-calorie ketogenic diets for weight loss, and not just in my research, but in outpatient clinical settings, I've been disappointed in terms of the long-term results. That again, where you have people restrict fat in an artificial way, they don't eat to satiety, but they can use willpower to use this type of diet to lose a whole lot of weight. But when you look at the results of studies where you look at 1 year or beyond, I have yet to see a place where a rapid initial, very low-calorie ketogenic diet results in lasting effects in a group of people. We all know individuals who've done this, but show me a group of people. And so we tried to pull together data from 5 published studies, and the yellow one at the bottom, the purple one, and the green one are all studies that used formula weight loss diets under 1,000 calories per day for roughly the first 3 to 5 months, and then were followed up from anywhere to a year to 2 years. And you can see that in all of these cases, by the end of the first year, weight regain is beginning. And then the blue line in there, I apologize to Dr. Sarah Hallberg, who'll be presenting data from the study she's heading up, working with us at Virta Health on reversing type 2 diabetes. And this is a case where when people are eating fat to satiety from day one, that the weight loss is slower, but it's more progressive. And at the end of year 1, we don't see weight rebound and weight regain. So because a chronic condition such as type 2 diabetes isn't going to be cured in 1 year, or isn't going to be cured in 6 months, from a philosophical point of view as a physician, why would I want to use something which is only going to provide transient benefit to the patient and then have them begin to lose control and gain that weight back? And so I would say that if it's a well-formulated ketogenic diet, you have to have demonstrated for that condition that it will have a lasting benefit for the average person for whom it is prescribed. And again, we've picked type 2 diabetes as our primary target, and we picked it in part because it's the hardest one to deal with, because when people have high insulin levels, so secondary to insulin resistance, administered insulin, or secretagogues, insulin secretagogues, those are the people who have the hardest time in achieving the metabolic changes. And so we think we're making progress in meeting this characteristic of a well-formulated ketogenic diet. And then the last two are, when people are on medications for hypertension or type 2 diabetes, many of those have to be taken away promptly when the diet is started. And you have to have real-time medication management through the physician and pharmacist or nurse and office staff in order to avoid the side effects of too much medication in the context of too little carbohydrate coming in from the outside and dramatically improving insulin sensitivity through metabolic pathways we have yet to define. And then the last one, and I thank Dr. Volek here, because when we came up with this list, he said, you got to put on number 10, and that is, a well-formulated ketogenic diet does not necessarily adhere to traditional dietary guidelines. Yeah. You get into conflicts with a number of guidelines when you try to maintain people's well-being and function on this type of diet. So my next to the last slide is, if I can get the right button here. The first is that needs to provide adequate sodium. And as I said, with the exception of hypertension or congestive heart failure or the other not all that common conditions where people have to restrict sodium, our experience clinically is we need to provide them with 5 grams of sodium intake per day, which is double our current guidelines of 2.3 grams per day. And we have a speaker, Dr. Andrew Mente, who has published seminal data in this obscure journal called the New England Journal of Medicine from the Pure study that demonstrates that for the populations around the world in different cultures, the low point in terms of mortality for measured sodium excretion indicating intake puts the optimum intake in the range of about 4 grams per day without people being in nutritional ketosis. Add nutritional ketosis, 5 is a modest number. Again, it's horrifying, but particularly when somebody's in maintenance, for the average, even a person burning 2,000 calories a day, they need to eat about 150 grams of fat per day. And the type of fat matters. That the, quote, healthy polyunsaturated fats, which are healthy if you're eating a very low-fat diet and you need those polyunsaturates as essential fats, isn't the same mix of fats that you should have when you're on a well-formulated ketogenic diet where most of your fats coming in are used for fuel, not for structural requirements for membranes and eicosanoid production and things like that. So that means avoiding the high polyunsaturated vegetable oils that are currently in the marketplace. And the demographics of that are changing. That I'm told that there's now a significant movement in the plant sciences area to move to a high monounsaturated versions of corn and soy, and it already exists for sunflower and safflower. As Jeff emphasized, that if you're in nutritional ketosis and you've doubled your rate of fat oxidation, it appears that some of the first fats that get into the pathway for oxidation are the saturated fats, and in spite of higher intakes, they don't build up in the blood, and if anything, are reduced. And so again, worrying about saturated fats is only associated when you're combining them with a significant proportion of carbohydrates in your diet. Again, getting adequate potassium and magnesium as minerals, those are important in whether you're on a high-carb or a low-carb diet. And on a low-carb diet, those come predominantly from leafy green vegetables and to some degree berry fruits. And if people are so motivated, they can make their own bone broth, which again is a source of not just adding sodium to the diet, but also providing those other nutrients. And the final point is, this is not a high-protein diet. And although we have Bob Atkins to thank for being a pioneer and a bit of a curmudgeon in terms of persisting out there, selling lots of books and making this a popular point, he did call it a high-protein diet, and he's forgiven for that. But we need to move on. And the last slide, again, was a slide that Jeff made. Do you want to describe this slide, or should? Anyway. And so when I started out in this field, my goal was, how do we mobilize fat? How do we get it to be burned? And if you mobilize it in the accelerated rate, pass it through the liver, make ketones, and use the ketones to feed the brain, I thought that's revolutionary. But of course, George Cahill figured this all out and published it in the 1960s. What's happened since then is we now understand that ketones are not just a fuel and not just for the brain. They appear to be an optimum fuel for heart function, and now there's considerable evidence in terms of gut function that if you eat dietary fiber and your microbiome makes it into butyrate, which is considered to be an optimum fuel for the gut, that's easily supplanted with beta-hydroxybutyrate. And if I make 30 grams of butyrate per day from 30 grams of dietary fiber, that's a lot. But my liver currently, I'm guessing, I haven't checked my meter, is making between 50 and 100 grams of beta-hydroxybutyrate a day. So again, this is a fuel for multiple organ systems, but now we know it's a signal. So you've got a molecule made in one organ, and it goes to other organs, has multiple effects, and so you have to think of this as a hormone. This is a very beneficial regulatory hormone that regulates the body's defense against oxidative stress and inflammation, which has an impact on longevity and lifespan, impact on inflammatory diseases, and cell signaling and mitochondrial function, including increasing mitochondrial NAD+ levels. So this is a potent signaling molecule that we are just scratching the surface on what it does, how it functions, and how do we modulate that, and how do we modulate it to the specific needs of individuals. Thank you very much.